I'd like to welcome you to today's Medical Center Hour, a program called Building Life, the Promise and Challenges of Synthetic Biology. I'm Marcia Day Childress from the Center for Biomedical Ethics and Humanities, and I'm pleased to welcome all of you to this program today. In May 2010, J. Craig Venter, the scientist who raced against the US government to sequence the first complete human genome, Venter announced that his Venter Institute scientists had assembled from scratch the complete genome of a bacterium and successfully rebooted it to life in a cell of a different bacterial strain. While falling short of actually creating new life, the Venter Institute's dramatic accomplishment brought new attention to the rapidly developing field of synthetic biology. Improvements in techniques for genetic sequencing and synthesis offer scientists powerful new tools with which to manipulate the basic building blocks of all life. Indeed, synthetic biology in building life versus simply taking its forms apart at the cellular or molecular levels holds great promise for helping us to better understand how life is constituted and how it performs. While this new technology may give us new and improved drugs and environmentally friendly ways to make renewable biofuels and industrial chemicals, synthetic biology also raises a raft of ethical concerns. Some of the more urgent questions have to do with biosecurity, biosafety, and environmental risks. Similar questions arose nearly 4 decades ago when genetic engineering was introduced. So whether there's anything really new and different about synthetic biology to warrant our exercising additional caution may be debatable. Nevertheless, President Obama's new commission for the study of bioethical issues made synthetic biology its first order of business and the subject of its first report, which was issued last December. Our Medical Center Hour today takes stock of synthetic biology, where we are and where we're going with the science, the anticipated applications, and any likely policy making to stimulate, constrain, or monitor it. Our speakers today come from within UVA. On my immediate right is Jason Papin, an assistant professor of biomedical engineering. On his right is Michael Rodemeyer, a longtime biotechnology law and policy analyst who now teaches in the Department of Science, Technology, and Society in the School of Engineering and Applied Science. Together, they're going to introduce us to the promise and the challenges of synthetic biology. We also have in the audience a member of the Obama Bioethics Commission, up in the far left corner, or for you, the far right back corner of the auditorium, John Aris, the professor of bioethics in the Department of Philosophy, and as I mentioned, a member of the commission, who I think will be willing to offer some comments when we begin the audience comment and discussion portion of our program. So now Jason Papin and Michael Rodemeyer and our primer in synthetic biology.
Thank you, Marcia. So Michael and I wanted to start off today trying to introduce a little bit, starting with some fundamentals of biology, just to make sure that we have kind of a foundation on which we can build to understand the context in which this new area of synthetic biology is developing. Thanks. And it's been coordinated. Thank you. So 1953 was the famous publication of the structure of DNA, and over the subsequent decades and years, many biologists have helped delineate some of the very fundamental principles that we now understand in molecular biology. As this graph depicts, within each cell, we have a nucleus that contains the DNA or the genetic content of that particular cell. This DNA is comprised of 4 base pairs with the letters here A, T, C, G that form common pairs where A binds to T, T to A, C to G, G to C as indicated here. Now each of these stretches of DNA contain a sequence of this ATCG in such a fashion as to correspond to particular genes, which when read correspond to individual proteins. These proteins are the workhorses of what happens inside of a cell. Now, just to give a sense of the scope of what exists inside of a human cell, we have about 3 billion base pairs in the human genome. So 3 billion of these ATCG sequences. There are about 20,000, maybe a little bit more than 20,000 genes in the human genome. And each of these genes get turned off and on depending on the environment, depending on the cell type. And there are about 200 different cell types inside each of us. Now, what's happened over the last couple of decades, we have improved, and engineers have made tremendous progress in sequencing or delineating what this particular ATCG sequence is within a variety of different organisms, within a variety of different cells. This process has been automated to a point to where we can buy a relatively small machine and for a relatively low cost delineate the entire set of millions and millions of base pairs in a particular cell. So with that advance, another advance that has come along the way is the ability to fabricate or actually synthesize these sequences of DNA. This fabrication process is developed where there are existing companies now where you can place an order, can send an email, I want ATTCGG, and they'll synthesize that particular stretch of DNA for a relatively low cost. I think the latest cost is something like 30 cents per base pair or something like that. Now, another big advance that's happened over the last couple of decades is the ability to model a lot of these processes. We have developed, and this is my own area of expertise, the ability to use mathematical computational tools to try to understand what's happening inside of these cells, which genes are being turned on, which genes are being turned off, and the consequence of turning these genes off and on. Now, with this ability to both sequence or understand these letters, these sequences in DNA, the ability to synthesize stretches of DNA from scratch, and the ability to model or try to understand the context of how these genes interact with each other, this field of synthetic biology has developed. Now, I lifted this definition from the commission report that came out just a few months ago. And, but to emphasize that in the field itself, many of the scientists involved have varying ways of defining this field of synthetic biology. But as kind of a working definition, just to kind of stimulate some of the discussion here, synthetic biology aims to apply standardized engineering approaches or techniques to biology with the goal of being able to create organisms or biological systems that have novel or specialized functions to help address a variety of needs. So I wanted to give just a sampling of some of the most recent publications and some of the most recent effort in this field. They span a variety of, from what I would say maybe are cool things to very applicable things. As one example, Jim Collins's group at Boston University published this paper a couple of years ago, Synthetic Gene Networks Account. One of the key objectives in this paper was to design a set of genes that together would result in some kind of counting mechanism inside the cell. As another example, as you see here, malaria is a tremendous disease. It affects 500 million people per year. And the current treatment, one of the most effective ones, is the use of this compound called artemisinin. It's extracted from sweet wormwood, depicted here in this picture, and it's one of the more current and effective treatments. However, the process of extracting this compound from this plant is very costly, very difficult. So in a synthetic biology effort, Jay Keesling's group at UC Berkeley and Lawrence Berkeley labs developed a process or engineered a strain of yeast that could produce this compound at a considerable reduction in cost. Now the basic idea was he started with a yeast cell. What you see here are the metabolic pathways associated with converting a simple sugar into a variety of different metabolites. And then he introduced from the sweet wormwood plant, isolated these genes and put them inside the yeast in order to add this additional pathway so that you could start in yeast from the simple sugar to produce compounds that yeast naturally produces, but then siphon off some of those compounds, undergo a couple other chemical transformations in order to generate this compound that can be used as an antimalarial drug. So this emphasizes, I think, a couple concepts to how synthetic biology happens. One, from a top-down approach where we start with existing cells, bacteria. We identify different processes in these cells that we find advantageous for some engineering objective. And then we can introduce those processes into yeast or some other cell that has perhaps some industrial utility. And these modified cells then perform some particular function that we have value, add value to. So this top-down approach is something that's really happened over probably a couple of decades. The beginning of biotechnology, which Michael will talk a little bit more about in a second, really underwent some of this process with the production of insulin. I think one thing that's really stimulated a lot of visibility in synthetic biology field is captured here in this bottom-up approach, where we start with this vast amount of information that we have on DNA sequences in a variety of organisms and assemble those sequences in some kind of order that corresponds to genes or some functions that we would want to try to create. And assembling these genes piece by piece until we create a system that has some engineering design. Now that was really what was at the heart of Craig Venter's approach that was published just this last year and stimulated the discussion from the Presidential Commission on Bioethics. This paper was published July of 2010, and just to kind of emphasize the title here, the emphasis was on the creation of a bacterial cell by a chemically synthesized genome. The objective was to create a genome from scratch and be able to create a cell from scratch. So just to emphasize the basic process here, they started out with a particular sequence that they wanted, and they assembled these nucleotides into relatively long stretches, oligonucleotides. These oligonucleotides were then assembled into larger stretches of DNA that were inserted into yeast. Then these longer stretches were assembled together to get even longer stretches until inside of yeast we had this complete circular genome. Once it was in yeast, that genome was then inserted into a cell that had its own genome removed. So I think this is an important distinction between creating just a novel cell and what Craig Venter did was he assembled a genome in the context of yeast and then took that genome and inserted it into another cell whose genome had been removed. So there was still these starting points of an existing cell in which the genome had been inserted. Let me now turn the time over to Michael to give a little bit of historical context to where Craig Venter has now ended up here in this synthetic biology.
So all of this is very interesting. It's certainly remarkable what scientists have been able to do in the last decade. But the question, in many respects, is, so what's the news? Is this something that's really new and different, or is this really simply an extension of the kinds of technologies we've been doing for quite a long time? And why is it even important to ask that question? Why is this new? So this is, of course, the classic half-empty, half-full glass of water. Certainly from one perspective, you could argue that synthetic biology, and as Jason says, it's really a collection of tools and techniques more than it is an organized scientific discipline at this point. But the question is, This has obviously been built on and made possible by rapid developments in technology, things like gene sequencing, synthesis, gene synthesis, and of course computational abilities through much larger and faster computers. All of those things have come along to allow synthetic biology to happen. And of course, if you look at the long history of genetic engineering and genetic tinkering on the parts of humans, you could argue, of course, that we've been working very hard to change the genetic basis of plants, animals, and bacteria for thousands of years, not often knowing what we're doing. But you could certainly argue that when you look at things like bacteria that produce insulin, or you produce soybeans that resist being killed by Roundup pesticides, or maybe even a chihuahua, you can argue that we're doing things that nature clearly would not have done left to its own devices. So this is really just another extension of technology that we've been already using to manipulate natural organisms for human purposes. And from that perspective, you could argue that there's really nothing arguably new about this technology and this approach. So the question is this really an Asilomar moment? An Asilomar was a conference held by the molecular biologist community, much smaller, back in 1975. After the initial discovery of the ability to cut and paste genetic sequences from one bacteria into another. That was really seen as a clear leap in technology that scientists actually came together to discuss the implications, both the safety implications as well as other concerns about this new technology before racing ahead to develop it. Because there was a lot that was uncertain and unknown about the technology at the time, scientists gathered in Asilomar, California in 1975, actually putting a moratorium on their own research until they could put some procedures in place to ensure that both laboratory workers and the environment would be safe. So is this another Asilomar moment? Is synthetic biology at the point where there is something new and different? So I've argued the perspective that no, this is really just a continuum of the kinds of evolution of technology that we've seen in many other areas. But from other perspectives, synthetic biology really does represent something fundamentally new. And there are a couple of points I want to make here. One is that up till now, we have largely, as genetic engineers, been able to reshuffle the deck of cards that nature has given us. Essentially taking the genetic sequences that nature has evolved over time and conserved and recombine those from one organism to another. So we're moving things that exist from one type of organism to another in our traditional recombinant DNA engineering. What synthetic biology permits us to do is not only to do that much faster and much easier, but in fact to bring our own deck of cards to play with. We don't need to rely on the deck of cards that nature's given us. We can create our own deck of cards. And in fact, those base pairs that we looked at earlier on, we really don't need to be stuck with those four chemicals. We've already created artificial DNA that uses different kinds of base pairs. So suddenly the palette that an engineer now has to look at life and to create new forms of life have gotten much broader. The tools are much more powerful than what we've been able to do through traditional genetic engineering. So we can look at the potential to create organisms and potentially, certainly, behaviors of things that really have not been seen before, that have not really had an analog in nature and in evolution. So we want to talk about why we want to do this, why we would want to have this kind of power. And Jason's now going to talk a little bit about some of the implications for applications that this technology may raise. And then I'll come back to the question of, after we talk about some of the promises, well, what are some of the concerns that have been raised about this technology? And is this idea of novelty an important issue for the public and for policymakers to pay attention to?
So the first application we wanted to emphasize is that of medical utility. As Michael emphasized, and as perhaps most of you are aware, insulin was perhaps one of the first examples where a synthetic biology concept really had play. That's the synthetic production of insulin. Another example, as I emphasized from Jay Kiesling's group, was the production of this antimalarial compound in yeast in order to bypass this process of extraction of this compound from leaves of this cornwood. Another example that's probably more immediate on the horizon is the expedited production of influenza vaccines. As we know, this process of generating influenza vaccines can take such a long time that often we have to predict which strains of the flu virus are going to come around in the next cycle, and then engineer vaccines that would correspond to that. The idea is that you could create libraries of different potential vaccines and then produce these at a much higher rate. Another class of applications in synthetic biology are with agricultural and environmental concerns. The oil spill that happened on the Gulf Coast this last year, there was a report that came out not too long ago that was able to evaluate the degradation of oil in the Gulf Coast. And surprisingly, there were particular bacterial species that naturally degrade oil, and there was a growth of this bacteria that helped degrade oil processes. The idea with synthetic biology would be to expedite this process, take advantage of some of those oil degradation or other pollutant degradation capabilities, be able to control that process and introduce it in particular environments. Another would be the enhancement of crop production. There are examples where strains of particular plants have been engineered to be insecticide resistant to a variety of different pests. Another example is to breed these animals or plants that are resistant to particular types of disease. So there's a variety of applications in this field. Another big area, and one in which synthetic biology could very well have its most immediate impact, is that of energy. To give kind of a concept of how biological systems can be used to produce energy, just think of the composting that you may do in your own garden. Throw a bunch of plants in there, a bunch of food left over from dinner, you close it off. After a certain period of time, when you remove that cover, you can feel the heat that comes from the bacteria and microbes degrading that organic material. That heat represents energy. It's enhancing this process that a lot of synthetic biologists are trying to do in order to address some of the energy needs that we have. So there's a big effort funded from the Department of Energy in order to generate or stimulate research in an area to develop microbes that can degrade plant feedstocks, plant waste, these kinds of material into ethanol and other compounds that have value and energy needs. And the last example I want to highlight here for energy needs is one that my own lab is connected with. That's the interest in generating algae that can produce hydrogen and other biofuels. Just to give a little context for this, this map represents geographical areas that are necessary or for generating sufficient fuel to displace the gasoline that's used in the United States. What you see in this box 2 represents the corn grain ethanol, the ethanol produced from corn grain at current production yields. The box 2 represents the amount of area that would be necessary to grow corn in order to produce enough ethanol to displace all the gasoline that's used. What you see in the box 4 is the total area of land that's devoted to producing corn in the United States, which was a surprising amount when I saw that personally. Box 3 represents the amount of area that would be required to grow switchgrass in order to generate enough fuel to displace gasoline. And in box 1 represents the amount of area that would be required for hydrogen production from algae and at a 10% solar efficiency in order to have that same energy content. So the idea is, can we engineer algae with this kind of potential yield that could produce enough fuel to serve a tremendous need that we have. So, as I mentioned, my lab's expertise are in computational models of these biochemical processes, and we've published some work where we've developed some models of an algae called Chlamydomonas reinhardti. It's a model system for understanding photosynthesis and lots of other processes. And the idea is, with this computational model, we can delineate all of the relationships between these compounds, these genes, these enzymes inside this algae cell. And with a synthetic biology approach, the idea would be if we want to convert sunlight, water, these basic sources into hydrogen, fuel, or biodiesel, can I identify which genes I would want to remove from this algae, or what genes might I want to add to this algae in order to expedite or make this process more efficient? So hopefully that gives kind of a sense of the scope of types of applications that a lot of people in synthetic biology are working to try to address. I want to give some time now to Michael to go through some of these ethical concerns that can help us place some of these potential advantages in context.
So we've looked a little bit at some of the potential applications of synthetic biology. It sort of gives us a good answer for the question of why would we want to do this. There clearly are some opportunities and challenges that synthetic biology may give us tools to accomplish that we can't accomplish in more ordinary ways. But what are some of the concerns, and what are the issues that are associated with this question of novelty that I raised before? One of the reasons that it's important to have a framing around this issue of novelty is in part because of public reaction. If you look back at the 30 years of experience with genetic engineering, while it's not that much of a controversy in the United States, the issue of genetically modified food is something that has literally created enormous trade barriers across certainly the Atlantic. In Europe and many parts of the world, genetically modified food is certainly seen as a technology that's unacceptable, that's socially unacceptable. And part of the reason for that is the framing issue. I think when we as citizens, as consumers, hear about some new technology, our first reaction is to say, well, what's this like? What's this like that I have some experience with? What should I think about this? And for many people, the argument is, well, if this is really just like things we've already been doing, then that's not something I need to pay much attention to. Back in the early days of biotechnology and genetically engineered food, for example, the biotechnology industry used to advertise that this is the same technology that we've been making beer and wine with for 10,000 years, so there's really nothing here to worry about. So it's a similar argument here. That, again, we've talked a little bit about some of the similarities. On the other hand, this is really new. Maybe we really have to pay attention to this. Maybe it creates some novel risks or novel concerns. So let's kind of walk through some of the issues that various people have raised as potential concerns about the technology. The first, and clearly one of the major issues, and this was certainly an initial reaction to the publication by J. Craig Ventnor of the new cell, is, are we playing God here? Are we actually taking on the role of creating new life? Now, I think for the time being, and I think the President's Commission agrees with this, that we've dodged that ethical bullet, because whatever we may think about the issue, that's not what Craig Ventnor did. But there may come a time, and it may not be that far off, where we actually do have a novel bacterial cell that has really not been seen in the natural world before, and not something that can be evolved on its own. And then the question becomes, where is there a line between the role of humans in using the natural order for our own purposes, and essentially usurping that divine role, if you will, and creating life on our own? You don't need to necessarily have a religious framework to address this issue. Of course, if you think about the secular meaning of this, This is Frankenstein, right? This is the hubris of the science, creating life in the laboratory and not fully knowing what the consequences are going to be. It's a trope that Hollywood knows very well. Every Jurassic Park sequel has this notion to it. So it has, yes, a secular meaning as well. Humans sort of going too far with the basic building blocks of life. So we haven't reached this threshold issue yet. And arguably, certainly for the foreseeable future, almost all of this work will be done on bacteria, on viruses, on algae. These are not generally organisms that we as a society provide a high moral value to. So this is not like Dolly the goat from now 15 years ago that shocked everybody when we realized that with the development of somatic cell nuclear cloning that Dolly was actually created a genetic clone of her parent. I mean, there were things that were shocking about that for a number of reasons. One was that, first of all, it wasn't supposed to be able to happen. Secondly, that it was this very cute goat, right, Dolly. And three, of course, it got immediately to that slippery slope about human sexual reproduction, which is an ethical and moral minefield. I think we're long away from that area in this technology. But if the technology develops as rapidly and successfully as many hope, we may not be far away from addressing some of those issues. Perhaps the more present concern, and it's one that federal officials and others have already dealing with, have to do with biosecurity. Back in 2002, scientists published the entire genetic sequence of an infectious poliovirus that had been assembled from scratch, from labs from chemicals in the laboratory to create an active infectious poliovirus. And again, all it consisted of was code. And it's available on the internet as a part of the scientific publication. A few years later, scientists published the genetic sequence for the 1918 flu virus, the one that was the super virus that killed millions of people around the world. And of course, again, it appears on literally just computer information disks accessible by the internet. And of course, the concern began being raised, well, aren't we just giving bioterrorists new tools simply to make more lethal and potent bioweapons? Thanks to the internet, everybody can now see what the 1918 Spanish flu virus looked like and potentially what made it so lethal. As a result of that, there have been lots of discussions in the federal level with the scientific community about sometimes what's appropriate to publish and what is not. But of course, the argument is, as it always is in science, that there are two edges to the sword. That the more we learn about what made that virus so lethal, the more we can be prepared to create the vaccine to prevent it from breaking out. And there's clearly a lot of work in that area, as Jason also recognized. I think the sense, again, of most people in the security community is that simply having the sequence information, which is in fact readily available, is really not enough at this point to give a new weapon to bioterrorists, because it actually requires a fair amount of sophistication to basically put this together and then to put it into a form that actually could be delivered. In addition to which, nature has already provided some really nasty candidates anyway, so why bother? So the sense is that right now this is not a concern, but again, if the costs and the ease, the cost decrease and the ease of doing this technology becomes rapidly disseminated around the globe, it's just as far away as a cable modem. The potential for this technology to get into the hands of many people is going to happen very rapidly. In fact, there's already every year, an annual competition where high school and college students are encouraged to build new organisms from a standardized set called biobricks that Drew Endy and his colleagues at Stanford have put together. Every year they come and they compete to see which team has created the coolest new organisms assembled from these standard building blocks. These are high school and college students. These are not sophisticated national laboratories making these kinds of organisms. So there is concern about how fast this technology will disseminate and who will have access to it. Biosafety, these again are not new concerns. So that there's nothing about synthetic biology that in and of itself creates unique risks. These are risks that we deal with in genetic engineering and in biosecurity. Biosafety, obviously laboratory workers and researchers have had to deal with the difficulty of dealing with microbiological contamination and infectious disease agents for a long time. The idea of having to handle potentially dangerous microorganisms in a laboratory is something with which we're very familiar. The notion here, again, is could you create some organism inadvertently in your laboratory that would end up having some lethal effects that could affect the health either of laboratory workers or potentially of the community if it were to escape from the laboratory. That's the concern that's been raised here. It's the Andromeda strain, Hollywood version for synthetic biology, but again, not particularly a novel concern. Things that the NIH has been regulating in terms of safety for biomedical research with genetic engineering for 35 years now, and is certainly looking at synthetic biology as well. The other category has to do with environmental issues. Some of the applications that Jason was talking about are talking about having organisms that have been synthetically created or created through this technology released into the environment. So for example, the algae that we're talking about would have to grow in large open ponds at quite a large scale in order to make enough biofuel to be relevant. What happens to the birds that come by and feed on those algae, for example? You introduce organisms into the environment, and we don't always confidently know what the implications of introducing organisms into a complex ecosystem could be. The good news is that certainly for most of the types of organisms that we're talking about, certainly in the initial level, these are all intended to be used in contained environments. And to the extent to which they are highly artificial, they're also highly unlikely to be fit for survival in the environment in the event that they would escape. Many scientists believe that the more artificial they are, the less likely they are to pose an inadvertent harm to the environment. Nevertheless, there's a lot of work going on to look at the idea of biological leashes on any kind of synthetic organism. So in the event that it does get into the environment, there would be a way to make sure that that organism does not reproduce or replicate in the environment. Again, these concerns have been around for some time. The same set of concerns have been raised for genetic engineering for almost 35 years. And by themselves, the issues are not new. What may be new about synthetic biology is that it introduces a new complexity and a new novelty that may make it difficult for both researchers and regulators to understand what those risks are in advance in order to ensure that they're handled appropriately. I think initially, the next couple of years, this is certainly not going to be an issue. The kinds of things that we're likely to see are going to be very similar to the kinds of things we've seen before. But as the technology develops, we need to be ready to think about different ways of doing risk assessments to make sure that the public health and safety and biosecurity concerns are protected. The issue is, for example, could you have emergent behavior from an organism from which you have assembled various pieces from various other organisms or created from scratch in a laboratory? Right now, we don't know enough to be able to predict with great confidence simply from the structure of the genetic sequence itself. This is an area where the President's Commission has also called for more research. The President's Commission looked at all of these range of issues and essentially said there really aren't new issues here. There's nothing novel about the issues being raised by synthetic biology. But as the technology continues to develop, we need to have a policy of prudent vigilance, that we need to be understanding whether or not we have systems in place, both internally within laboratories and from products that may be developed, to ensure that these products are used and developed safely. The Commission noted the existence of the current regulatory system for biotechnology that's been in place for 30 years, which really is not a comprehensive system, but a patchwork quilt of existing regulatory agencies and authorities that regulate genetic engineering products. For now, that system is probably adequate to deal with any concerns that may be raised about biosafety or the environment. But again, looking ahead as the technology develops, it's important to understand whether or not we have the regulatory framework in place. Finally, one other point I want to raise, which is something that is kind of unique about synthetic biology, which is the rise of what is called the do-it-yourself community. This is really exemplified by this international competition where you have high school students and college students under the supervision, hopefully the adult supervision, of their university participating in this international collaboration contest. But we know that there are a number of people, particularly in Silicon Valley, sort of wealthy hobbyists, who have said, this stuff is pretty cool. And you know what? It's a lot like the information technology that I'm already very familiar with, manipulating large-scale models, a lot of information on the computers. These are things I can do in my laboratory at home. The barriers to entry to doing this are very low and are continuing to fall. So right now, while you're unlikely to be able to do a lot of the very sophisticated synthesis that certainly Craig Venter and a few other labs have been able to do, down the line, that may begin to change. And of course, no one regulates what people are doing in their garage. No one is really paying attention to, these people may be outside the institutional framework. And yet, potentially some of the risks that they could inadvertently introduce are no different than the kinds of risks that might be introduced by researchers in a laboratory. Again, this is an area that the President's Commission engaged with the do-it-yourself community to try to figure out, are there ways to encourage this to continue to happen without making sure that, by also making sure at the same time that safe practices were followed. And again, this is, I think, part of the Commission's idea that we need to watch as this technology begins to develop. Again, for now, not an issue, but a few years from now, it could grow into something where we might need to be thinking about ways to look at or regulate, perhaps even on a local level, these kinds of activities. So Jason and I have tried to give you a little bit of an overview of both the promises and the potential perils of synthetic biology and some of the debate that I think you're likely to see over the next few years as this technology continues to develop. And I do believe it will continue to develop rapidly. With that, at least I hope you'll be better forearmed and forewarned.
Thank you both. This was a fascinating presentation, and I think we have plenty of time for your questions, comments, and discussion. And I'd love to give the microphone first to John Aris, again, the Porterfield Professor of Biomedical Ethics here, but also a member of the Obama Commission.
Thanks, Marcia. I want to thank you both for really succinct, clear, helpful presentations this morning. You've been talking about how synthetic biology has been built on previous decades of work in molecular biology, but I would add that previous ethical and regulatory work has been done by other bodies that the Commission leaned very heavily on, in part Michael's excellent work with the Woodrow Wilson Center. Seems to me that one interesting way of looking at synthetic biology is as a kind of Janus-faced phenomenon, right? So on the one hand, it's very corporate-oriented, right? I mean, as soon as Craig Venter published his results, you know, he was packing them under his arm and taking them down to the patent office to you know, try to get patents on all these techniques. So one problem with the corporate phase is that it does raise interesting problems with regard to, you know, the public benefiting from these discoveries, right? In terms of erecting very high patent barriers to access to this information. So that's one side that you might want to comment on briefly. The other side is the sort of anarchic wiki sensibility that Michael referred to under the heading of, you know, do-it-yourself biology. And here, we're not so much worried about playing God as we are about just playing. Okay? So as Michael was suggesting, it's now going to be possible for people to download all this information off the internet and just play around with it in their garages. Now, one problem is that, and you've really alluded to this already, Michael, which is that in the past, the Asilomar moment, involved university researchers or corporate researchers who have a university history, all of whom have been pretty much enculturated, hopefully, into a culture of responsibility, right? So I'm now wondering what's going to happen when you have this whole wiki crowd of do-it-yourself biologists doing really fascinating, interesting things. I just saw a video of Drew Endy on the web where he was talking about taking bacteria from the big toe of a biologist and making cheese out of it. and one of the winners of the student competition, the IGEM competition, took E. coli bacteria, and if you've worked with E. coli, you know that it really smells terrible. And these students jerry-rigged the E. coli to smell good, and they called it O. D. coli. Okay, so I just wanted, so if you could take either one of those prongs and talk about, what the corporatization of synthetic biology might imply for justice issues or access issues. And then there's the other question about how are you going to regulate, and this is the question Michael already asked, how are we going to regulate people who don't come out of this sort of ethos and institutionalized ethics that you find in universities and even in corporate America? Thank you.
You guys want to take a stab at this?
Sure, well, let me start. There is a huge debate going on in the research community about the whole intellectual property issues. It has been imported wholesale from the information technology universe, the battle of open source technology versus the Microsoft model. That debate has been incorporated wholesale into this debate. So you have people like Drew Endy, who have created this BioBricks Foundation that are putting these basic components onto a publicly available registry without any kind of patent protection at all. These are and his notion is that this technology should be developed openly without patent restrictions, much in the same way that the sort of open source information technology movement has thrived. As you can imagine, there are other interests, and Craig Ventner would be one of them, who completely disagrees with that model and says that the only way that we can get innovation in this area is to attract funding from research, from capital venture funds, and the only way we do that is if we have intellectual property, because if everybody can just copy what we're doing, then no one's going to pay us any money to make anything. So that battle will go on. There is not, I think, any broader interest in the administration or in Congress to revisit the whole issue of what kinds of property can be patented and what not, and what cannot. And I think that what you're seeing is going to continue to see kind of a cultural clash between these two very different approaches to the development of technology.
And I just wanted to make a comment on this IGEM competition, just kind of give a flavor for the culture of that group. I've been one of the faculty advisors for UVA's team for the last few years. And the competition's international, and the students love it because it feels like a rock concert, actually. At the award ceremony, they actually play rock music really loud as everyone gets in there. It's a very unusual setting. But in this competition, every team will, many of the teams will have mascots dressed up as little viruses or all kinds of things. And I think, you know, there's both good and bad to that. It stimulates a lot of interest in the students to just, science can be fun. We can make a difference in trying to really engineer new ideas that can be used to address all kinds of needs. But on the flip side, I think it can maybe introduce a certain flippancy to the whole process. I think the competition does do a decent job, and every submission from every school is required, or rather expected, to have some discussion about ethical issues. Every school will try to come up with new ways of dealing with some of these. UVA's team, for example, one year put together a brochure that kind of outlined and highlighted some of the ethical concerns that the public should be aware of, trying to educate the public and using that form to do so.
Thank you. Other questions, comments?
My name is Tad Hardy. And Professor, you mentioned corporate responsibility. And I know that concerning the Monsanto Corporation, there are many people who have accused them of some pretty strong-arm tactics surrounding their genetically modified Roundup-ready seeds, which have actually resulted, at least in some countries, in decreased food security because smaller farmers who had been raising their own food for their local communities are no longer able to do so, and that seems like a very worrying trend to me, or potential trend.
Well, I think the President's Commission looked at that because there were the issue about how do you ensure that the fruits of technology are shared, that there's some equity, and both in terms of the risks are not falling on a particular population, but that the benefits are also broadly distributed. That's often where some of this discussion about patenting got involved in. I think if you look at some of the controversy about genetically modified food and really dig to the bottom of that, you will find that much of it really has to do with the behavior of the Monsanto company and certain practices that they chose to do. What I think is interesting here about this technology is that instead of the very first technology that we're likely to see as a product of the technology being a commercially proprietary product that can be used by very large farmers to grow food more efficiently in the industrial agricultural area. So it's things that had no benefits to consumers. That the first product here may be a malaria drug that will have probably, and could have potential real benefits to third world citizens who suffer from malaria. It will not be a benefit necessarily to the developed world. So I think it will be interesting. Again, it's sort of a framing exercise. We may end up seeing this technology in a different light than we saw genetic engineering, because the first applications may likely to be ones that have much broader public benefit than what we've seen in the area in agriculture.
Jim Childress in Bioethics. Let me thank both of you very much for wonderful presentations. And you were very thoughtful in considering pros and cons. But up on the screen we have the President's Commission approach of prudent vigilance, which has come under sharp criticism from those who say what we really need is a precautionary approach or use of the precautionary principle. I'm wondering if both of you came down on the side, given your measured presentations, of the prudent vigilance, or whether you think we need something more like the precautionary principle.
I'm not claiming a particular side here. I think prudent vigilance is probably where I would side, because among the points that Michael brought up are that most of the ethical issues are ones that we've been dealing with for several decades. And so I think there is a lot of the concerns that came up with the genetic engineering have provided a framework for us to evaluate the synthetic biology advances that are coming out. So I think because of that, the tremendous benefits like this production of the antimalarial drug is just one example, provide enough motivation to definitely push forward in the field. And because we have this historical context to evaluate some of these concerns, that's probably where I would personally side.
I think the challenge is this issue of uncertainty, which I talked about before, and how do you proceed in the face of uncertainty? There is the precautionary principle, which can be interpreted in a variety of ways. In its most extreme form, would say, well, you really can't go ahead in the face of uncertainty, which of course then begs the question of, well, how do you ever resolve the uncertainty? So I think that the way I interpret prudent vigilance means that we have an approach of learning stepwise as we go, that we learn in controlled environments, and we take risks that are controlled and limited, and we learn as we go along. And I think in general that's actually been what we've done in fact with genetic engineering. The initial restrictions back in the 1970s, many of the concerns turned out to be unwarranted, but we only learn that by doing. So it's not a green light, it's not, go out and do whatever you want and God bless you. It's really more of a notion that there are concerns and we need to be careful, but let's keep going so that we can learn as we go along.
There's another comment, just real quick, there's another comment in the Presidential Commission's report that spoke to the fact that, the field isn't really at a point yet to where there are significant ethical questions. Those are foreseeable, those could be in the near future, but because the Commission tried to evaluate some of these concerns a priori rather than reactively, I think in stimulating discussions in the media and the public in settings like this, I think it's making the general public aware so that when those concerns do come up, people are more ready to address those.
My name is Fred Hayden. I'm from the Department of Medicine here. I have two questions, one of which relates to the discussion you just had, and that really is practically what's been the worst abuse that you've heard about to date, and pragmatically, what's the monitoring system going forward to try to limit risk? And then the second question I have really relates to discussions going on in the public health community right now, particularly WHO in the process, with the intergovernmental process that is looking at the Convention for Biological Diversity and virus sharing, benefit sharing issues around particularly avian influenza response, but more generally. And I didn't know how that discussion would relate to synthetic biology going forward.
So the most significant abuse to date, I don't know. I don't focus on the abuses too much. I don't really know that I have a good example. Most of the visible items, I don't know, maybe Dr. Ayers, maybe you have an example of that?
The field is so new, there's just hardly a track record to go on.
Yeah, I don't think there's anything in this area that we've seen yet that would rise to that level. I think there are examples, if you look at the genetic engineering analog a little bit, clearly the problem that we've had there, one of the problems that we've had there, has been when you put these things in crops and plants, things happen. They move. Pollen moves. And it's become very difficult to keep genetically engineered crops segregated from other crops, organic crops. Most of those are economic reasons that people want that segregation to happen. They're not really environmental health or safety reasons. But nevertheless, there's a market that wants some of this product to be genetically engineered and others to be not. And there's no real way that can happen. There were instances in that area where we were looking at genetic engineering to grow pharmaceutical chemicals in food products. So there was, in fact, a corn plant that had been engineered with some human contraceptive that was being proposed to be planted and field tested in California. And I think it had received regulatory approval, but the press got a hold of this, and as you can imagine, the notion of corn that would make you sterile was probably not such a great product. So that was an example of something that probably ended up being somebody thinking in the laboratory, oh, isn't this cool? Look what we can do. We can use this corn to grow a lot of this stuff. And nobody really sort of sat back and thought about the bigger picture and saying, probably not a good idea. So that never happened. But that's an example of somebody coming up with a bad idea that could take place.
You have to comment on availability of the data, or
Yeah, well, is there anything, who's watching this process evolve in terms of trying to avoid situations like you just described?
like this? Yeah.
There's no central place that's looking at this. This is all, it's a lot of university research. It would come under the
Expectations and publications and making data available, Food and Drug Administration,
scientific conferences, that sort of thing. There's really not, nobody is really monitoring this. And of course, under the NIH, you have the system's institutional biosafety committees that would have to look at, within each institution, research that would fall under the guidelines, the recombinant research and some of the synthetic biology guidelines. So there are NIH guidelines that would apply to a lot of this research right now.
How about the marketplace is watching?
And, well, that's right.
I'm so sorry, we're going to have to close this discussion because we're out of time, but I think we're leaving with lots of questions. Marketplace, uncertainty, who's watching. I'm certainly left with a little nagging thought in my head of, will I see my grandchildren playing with bio-Legos? Sounds like they might already be here. But thank you both so much for a very stimulating and informative discussion.